Why don't we go get started? So we've made it through a whole year. This is the last uh, seminar of the seminar series this year. So we've all made it. So there will be no more for the rest of the summer. So I also want to thank the students for attending, even though you're required to. You asked some really good questions. I've been really pleased on how well you guys have asked some just actually shockingly good questions. I'm like, oh wow, I should have thought of that one. So anyway, as our last speaker of the year, we have Dr. Martin Darvas, who is an associate professor in our department, Department of Laboratory Medicine and Pathology. He is part of the Neuropathology division, so he's visiting us all the way from Harborview, as opposed to being nice and local. Martin got his PhD in biology in the Department of Molecular Psychiatry in the University of Bonn in Germany. And that's kind of a love of the molecular psychiatry is actually he's carried on throughout the rest of his career, which he'll talk about today. He then moved over to here, where he was a postdoctoral fellow in the Department of Biochemistry under the mentorship of Richard Palmeter, who was also really big into molecular basis of psychiatric neurodegenerative diseases. He's very entwined in the neuropathology research and has been focusing very heavily on neurodegenerative diseases, specifically Alzheimer's disease, which he's going to talk about today.
All right, thank you, Scott. Hello, everybody. So what I'm going to talk about today is an interesting conversation that I had with a colleague at UW, Warren Ladiges, who's in comparative medicine, and Warren has been studying aging for ages. And it was a mix of that and then the notorious cesium spill and the pandemic, which brought people together to talk about interesting projects. So we submitted a proposal to the NIH, and we actually got it funded. And the idea is to target Alzheimer's disease with a geroscience approach. Today I'm going to talk about a little bit about the geroscience concept in Alzheimer's disease, about a new approach for an Alzheimer's disease intervention, about an improved approach to capture cognitive, but also behavioral psychological symptoms of dementia in mice, and also a new approach to model Alzheimer's disease in mice. So first, a little bit about Alzheimer's disease. So it's the most common cause of dementia. You can say could be up to 70% of all dementia cases could be Alzheimer's disease. It's the 6th leading cause of death in the US across all ages, and also for ages bigger than 65. And for those above 85 years old, it's the 3rd leading cause of death. And the biggest risk factor for Alzheimer's disease, for sporadic Alzheimer's disease, that is age. So this is not a disease that afflicts the younger population. It does so mostly when you have autosomal dominant mutations in your family, and that is pretty bad. And you can see here on the right-hand side a projection of how the number of people with Alzheimer's disease, which is believed to be now at somewhere between 5 and 6 million people, to go all the way up to 50 million people by the year 2050. So a lot of people in this room will still be here, and a lot of people in this room will have to deal with this. This is just for the US. Take into consideration places where you have botched population politics in place, policies in place that result in a dent in the age distribution in the population, like the one-child policy in China, for example. They're really up for a rude awakening in 20 years or so, I would say, just based on this. And there is currently no cure available for Alzheimer's disease. There are treatments, antibody-based treatments that everybody is talking about, lecanemab, But that's not a cure, really, and it's a little bit early to be too optimistic, and it's also very, very pricey. So I would say, for the time being, hold your horses with that. So the search for intervention kind of like constitutes the El Dorado for Alzheimer's disease, something that everybody wants to do, because you can imagine that with Alzheimer's disease, if it's 6 million people that are affected, if it's really a large proportion of the elderly, you can imagine what this does to the caregivers, to the loved ones, and to the overall healthcare system and to the economy. Geroscience. So we are here at the intersection of basic aging biology, chronic disease, and health. So you can see on the left-hand side are concepts coming from aging biology, and then we have the aging physiology, and where you basically try to say, well, aging isn't a disease, it's just the inevitability of life, but a major risk factor for several chronic diseases and conditions, not just Alzheimer's disease, other neurodegenerative diseases, heart disease, what have you. All of these things don't get better with age, and especially neurodegeneration is something that is usually associated with age. And Geroscience seeks to understand the genetic, molecular, and cellular mechanisms that make aging a major risk factor and driver of common chronic conditions and diseases of older people. So this slide comes straight from the NIA Division of Aging Biology to really demonstrate how these things may be conceptually connected. And you can see that there are two keywords here. One is dementia and the other one is neurodegeneration. And I really would like to focus on these two here. So how does Alzheimer's disease fit into the overall processes of biological aging? So first of all, we know that there are Alzheimer's disease associated vascular alterations inside and outside the brain. There are diseases of blood vessels, cerebral and systemic blood vessel. They often coexist with AD neuropathologic change. A lot of times Alzheimer's disease is not a monolithic disease. It's in a co-pathology, and one of the strongest co-pathologies is cerebral vascular disease. Arterial stiffness is actually considered a risk factor for dementia. Midlife hypertension is recognized as a risk factor for dementia and Alzheimer's disease. There is cardiac amyloidosis in AD, and some of the AD genetic factors have also been linked to heart failure and dilated cardiomyopathy. And if you want to find out more about this, I have put some references at the bottom of the slide, and they're always at the bottom of the slide so that you can, I hope, find more information about things that interest you that we couldn't go over with enough time here. Another thing, which is kind of like, heart disease is one of the hallmarks of aging. Another one would be hyperglycemia and type 2 diabetes. And these may also be a risk factor for dementia. You can see here, this is taken from a study on the left-hand side that was done in Seattle, the Adult Changes Thought Study. It's a longitudinal study of aging in humans. And on the right-hand side, you can see a meta-analysis where you can see that there is a association, a risk, a hazard ratio for diabetes and dementia, and it's broken down by all dementia, vascular dementia, non-vascular dementia. So these studies here cover around 2.3 million people. So this is some very intriguing evidence suggesting that hyperglycemia and type 2 diabetes mellitus, they could be considered risk factors for dementia indeed. Well, one of the hallmarks of aging, of course, is oxidative stress. And there's a good amount of data suggesting that cellular beta amyloid, one of the major proteins that aggregate in Alzheimer's disease and aging, attacks the electron transport complex 4 and some of the key Krebs cycle enzymes. And the other major pathologic protein in Alzheimer's disease, tau, phosphorylated tau, can also contribute to inhibition of complex 1. So linking Alzheimer's disease clearly to some other of the very basic processes of aging. Another strong component of aging is what's considered inflammaging. So Alzheimer's disease has a strong inflammation component. It was just 1 year ago where there was a seminal publication coming out from David Holtzman's group, where he clearly demonstrates that in order to have a tau pathology-mediated loss of neurons, you need to have an involvement of T cells and of microglia, both adaptive and innate immune system components. Without those, you don't really see that much neurodegeneration caused by this Alzheimer's disease pathogenic protein. So we also know that there are late central nervous system inflammatory processes in Alzheimer's disease. And there are a lot of the GBOS variants in Alzheimer's disease. Probably most of them are found in cells that are part of the innate immune system, microglia and astrocytes in the brain. TREM2, ApoE, these are things that are expressed by these cell types. So late onset Alzheimer's disease, that's the type that we are talking about all along, not the familial Alzheimer's disease. Involves numerous mechanisms in line with processes of biological aging. I've given you a few headlights. The literature is chock full of more examples. You will find them relatively easy. So basically, the model is you have environmental factors, you have age, you have genetic factors, all conspiring on these key mechanisms here in the center of this slide to then ultimately conspire to result in what we call late onset Alzheimer's disease. That would be one way of really looking at it and of gauging Alzheimer's disease risk. So our hypothesis for this project is that targeting multiple aging processes associated with Alzheimer's disease, like vascular changes, glucose metabolism, DNA repair, et cetera, will alleviate behavioral dysfunction and also neuropathologic changes in an aging Alzheimer's disease mouse model. So one of the drugs we're using is a pretty commonly used drug. It's rapamycin, sirolimus. It has been shown to target multiple aging-associated processes. And on this slide, there may not be all the processes that you want to see or all the processes that you'd have seen because their number is legion. So rapamycin is marketed under the name Rapamune. So the drugs that I'm going to talk about today are all FDA approved already. And that's the whole purpose of this, to really use FDA approved drugs and repurpose them for Alzheimer's disease. And there are a couple of brain-related clinical trials for ALS, MS, and for cognitive impairment, alcohol abuse, epilepsy, and depression. It was approved by the FDA 25 years ago. And one of our former colleagues, Matt Kaeberlein, has actually performed multiple studies investigating the mTOR pathway all the way up and down. So you can see here on the right-hand side are just a few of the targets that are affected by rapamycin: mitochondrial dysfunction, proteostasis, genomic instability, stem cell exhaustion, epigenetic alterations, cellular senescence, telomere attrition. All of these kind of like funnel through this mTOR signaling to have impacts on Alzheimer's disease, vascular dementia, normal cognitive aging, dementia with Lewy bodies, and another type of dementia, and so-called mixed dementia, which is more than one form of dementia. And it has been shown that dietary rapamycin reduces vascular defects in old mice and also in mutant mice carrying the Alzheimer's disease risk allele. You can see here on the left-hand side that you had a reduction of large arterial elastic stiffness in old mice after only 2 months of treatment with the drug. On the right-hand side, you can see there's an improvement in cerebral blood flow in mice that carry the Alzheimer's disease risk gene ApoE4 after 6 months of treatment in the hippocampus. The next drug would be the alpha-glucosidase inhibitor acarbose. It reduces the postprandial blood glucose, and it has been shown to increase lifespan. You can see here on the left-hand side is the lifespan enhancement by acarbose in male mice. Interestingly enough, that didn't pan out so much in female mice. Cannot really say how. And on the right-hand side, you can see as a proof of concept the amount of glucose in the blood in these mice after treatment with different levels of acarbose. Our colleagues in the department have recently published this paper showing that acarbose also suppresses symptoms of mitochondrial disease in a mouse model of Leigh syndrome. These are NDAS4 knockout mice that were actually made by Richard Palmiter here in biochemistry. And acarbose reduced brain lesions in these mice. You have to know that these mice really don't get old. This is not a disease of aging. These mice die young because of these mutations. But it clearly increases survival, delays onset of the disease. It doesn't stop it completely, but it remodels the microbiome in its metabolism. And it has been shown that it even works better when it's given together with rapamycin. The last drug I would like to introduce you to is phenylbutyrate. That's an orphan drug approved for the treatment of hyperammonemia in patients with urea cycle disorders. It's an HDAC inhibitor. Basically, that's what it does. It acts as a chemical chaperone and inhibits ER stress. It has been used in phase 1 and phase 2 clinical trials for tumors, astrocytoma. It is marketed under the name Bufenil. And together with tauroursodeoxycholic acid, it's used in a phase 2 clinical trial for Alzheimer's disease. It's also used in clinical trials for Parkinson's disease, ALS, and Huntington's disease. And it's currently being tested in preclinical mouse lifespan studies. So the idea is, let's target multiple components of the pillars of aging, overlapping, admittedly, but let's just target a lot of them all together and mix it all up and see if we can make these mice healthy agers and thereby strengthen them when they are challenged with an Alzheimer's-like situation late in life. So the next thing that I want to touch on is the neuropsychiatric syndrome in dementia. And this is something that has been already observed in very, very early years of Alzheimer's disease history. So 120 years ago, Alois Alzheimer first saw Auguste Day at a community psychiatric hospital in Frankfurt. And the thing that the husband of the afflicted individual said was that she suffered, that she had an untreatable paranoid symptomatology. And then pretty fast, she got sleep disorders, memory disturbance, aggressiveness, crying, and progressive confusion. And this patient was then later on analyzed on autopsy. And this is basically the first patient characterized in medical literature. And after the doctor who described this patient, Alzheimer's disease is named. But I want to point to the fact that one of the very first symptoms that this patient actually had was not what people usually associate with dementia. So how often do we see neuropsychiatric subsyndromes in patients with dementia? So here I brought out the big guns and show you a couple of large patient cohort studies from Europe and the United States. And you can clearly see that a lot of the clusters that are found in the neuropsychiatric data for these patients are around affective symptoms, apathy, anxiety, depression, psychotic and manic behaviors, vegetative symptom. I mean, some of these things may seem a little bit contradictory, right? You have apathy and you have agitation, but they have all been observed in patients with dementia. So they are part and parcel of dementia. So when you talk to people about Alzheimer's disease or dementia, and you ask them what afflicts people with Alzheimer's disease, a lot of times the answer that you'll get from them is, oh yeah, it's a problem with memory, it's a problem with thinking or decision-making, but all of these problems are highly prevalent. They're usually there as well, and they're causing a lot of problems for caregivers, and they actually conspire with the cognitive symptoms to really reduce the daily living abilities of patients with Alzheimer's disease, resulting in them having to be no longer being cared for at home, which of course is the worst thing that you can have for somebody that you take care of. So what are the mouse models that we use to study behavior deficits in Alzheimer's disease? So I've been talking a lot about late onset Alzheimer's disease. So instead of using the usual Alzheimer's disease models that have been developed over the last 20, 30 years, where you take mutations that are found in families where Alzheimer's disease has an early onset, is very aggressive. There are familiar mutations in genes like APP and presenilin. We take a different approach. We're using a so-called late onset Alzheimer's disease risk models from the Model AD consortium from the Jackson labs. And these are mice that have one risk factor for Alzheimer's disease, the single strongest risk factor with a high abundance, ApoE. If you carry one copy of the ApoE gene, your risk of getting Alzheimer's disease is around 4 times higher than somebody who doesn't have an ApoE variant. And if you have 2 copies of that gene, your risk may be somewhere between 8 and 12 times higher than people without the ApoE4 allele. And these mice also have a mutation in their APP gene, such as that the mouse part, which is very different when it comes to the A-beta-42 peptide, which is part of these amyloid plaques that are pathognomonic for Alzheimer's disease, that they're humanized. So if all the right factors conspire, these mice actually could develop plaques or could have detectable A-beta-42 levels similar to those in humans. The problem is they don't. So what we know about these models is there is an age-dependent increase of insoluble A-beta-42, but there are no plaques. So plaques are one of the pathologic hallmarks of Alzheimer's disease, and these mice simply don't have plaques, which is okay because it's just a risk model for Alzheimer's disease. It's not that these mice need to have plaques in order to be really a model of Alzheimer's disease. They're a model of Alzheimer's disease risk. We know that the targeted replacement of mouse ApoE with the humanized ApoE4, 2 copies of it, alters fear learning, neuronal activation, and it causes some baseline DNA damage in the hippocampus. What is not clear is exactly what happens if you bring these 2 mutations together. The Jackson lab is offering a strain from their mouse catalog, if you want to say so, that has these 2 mutations and also a mutation in the TREM2 gene, which is a little bit problematic because that TREM2 mutation seems to occlude a lot of the effects or dominate a lot of the effects seen in these triple mutants, which is why we decided to simply go with a very easy model, humanized ApoE4 and humanized APP. And the mouse behavior tests that we chose to model not just the cognitive, but also the neuropsychiatric complications in Alzheimer's disease were chosen such as that we can apply them repeatedly in a longitudinal setting, which is why you don't see things here like fear conditioning or water escape-based tasks, which are rather aversive and could add the element of chronic stress to the study. So to measure cognition, we used the Y maze to look at spontaneous alternations. A Y maze is exactly what it sounds like, a maze in the shape of a Y. There are 3 arms, and you can do perfect alternations. You go from one point to another and then to another and have a series such as that you don't visit the same arm twice in a row, so that you always do perfect triplets of arms 1, 2, 3, 1, 2, 3, 1, 2, 3. So that would be the ideal alternations situation here. And people have used this as a measure of attention processes and working memory. Apathy, we measure 2 different types of apathy here. One is nest building. So that is a goal-directed behavior. That's a daily living ability that mice have to do. If you give a mouse material to build a nest, they will. And then you can score the quality of that nest and use this as an index for this goal-directed nest building behavior. The other one is the more reactive behavior. Mice have a fur coat and they got to groom it. So if you spray a little bit of sucrose onto their coat, they will groom it. And then the question really is one, if the mouse is apathetic, it may simply not be interested in taking care of themselves anymore and not groom that much. For anxiety, we use 3 different tests. We use the open field test, which is one of the standard tests. It's basically, as the name implies, an arena that has simply an open part, and mice are afraid to go into the open. That's where they will be most at risk for predators. And they will venture a little bit into the open because they're curious animals, and most of the time they will, however, spend at the periphery of this arena where they can make contact with the wall by their whiskers and where they can stay safe. The other one is the elevated zero maze. It is actually that. It's elevated, but it has 2 parts of the maze that are completely open. So you make it a little bit scarier by not just having open spaces, but by adding the element of height to this. And again, a mouse will eventually go into the open, but they really try to avoid the open area. And a mouse that has anxiety will do more so than a mouse that doesn't. And the third test is the novelty suppressed feeding test. Mice are really, really reluctant to eat in a new situation. All kinds of things could be wrong. So if you have a mouse that is hungry because you fasted it overnight, and you put it into an arena where you expose it to some food, it will eat the food, but it will take the mouse some time to overcome the anxiety to consume it. These are all different levels of anxiety. You induce anxiety by usually overcoming a competition between the urge to explore and the fear of what might happen in open areas or in uncertain areas. So the first set of data will be about comparing mice that have the ApoE4 allele, which are the Alzheimer's disease risk mice, with the appropriate controls, which are mice that have 2 copies of the ApoE3 allele. So these also have humanized ApoE, but instead of having the risk allele for ApoE4, they have what is kind of like considered the basic, the standard against this risk is compared, and that's ApoE3. And we look at mice at 6, 12, and 18 months of age. And we perform this battery of tests that you can see here on the right-hand side, and you can see some illustrations of how these tests are designed. And then at around 22 months of age, there's a drug intervention where the animals get either a control chow, which is very similar to the medicated chow, or they get a medicated chow, which has compounded into it rapamycin, acarbose, and phenylbutyrate. And then we test at 24 and 27 months of age while these mice are still undergoing continued treatment with the intervention. So what we are getting out of this is longitudinal data of mice that are not exposed to a drug all the way up to 27 months of age, right? If you take the control mice. And after, for the last 2 timestamps, you get the effects of the intervention. So what does this look like with nest building? So if I lump male and female mice together in these graphs, then that means we tested statistically whether there were effects of sex on the behavior, but didn't find any significant main effects of sex. So as you can see here, in nest building, there was no difference between males and females. And yes, there was an effect of age. It was rather modest, and actually it went up, suggesting that these mice, when they're very young, don't really know how to make their nests. And once they get older, the nest building ability between 12 and 27 months of age kind of like stays the same. The next apathy test is the splash test. The way this goes is that this is video recorded, and we have an army, not an army, we had 2 undergraduates scoring these. And they're not done yet. We have gotten to the point where we analyzed all of these mouse videos up until 18 months of age. So there's still a backlog of these. But what you can see here is that the amount of time that the animals spend grooming themselves after they've been splashed with a sugary solution simply goes down with age. So yes, there's an increase in, if you want to say so, reactive grooming as an indicator of apathy, a decrease in reactive grooming, which we use as an indicator of increased apathy in aging mice. We observed significant main effects so far of age, sex, and genotype, which will make this really difficult to interpret later on when we add the other data sets, especially drug interaction. Anxiety, again, we saw significant effects of age, but also of sex, which is why we keep these together. We didn't see any genotype effects, unfortunately, which basically means that all mice had increased levels of anxiety in the open field and also in the elevated zero maze, meaning that these mice venture less and less and less into the open area of these mazes. Mind you, we spice it up a little bit, and every time these mice are exposed to these mazes, 6 months after they were exposed to it the last time, we add some olfactory cues, some odors, to make it interesting so that these mice are not simply saying, yeah, been there, done that. That's an important confound that we're dealing with here. And if you look at novelty-suppressed feeding, the latency to engage with food, young mice, well, it takes them around maybe 5 minutes and then they sniff it out. But if you look at the older mice, it almost takes them like 500 seconds or so until they really collect the will to go and engage with the food. So whereas with the open field, the reduction in time is an indicator of anxiety, here the increase of time is an indicator of anxiety. And we see a very nice effect of age on this as well. So we can clearly say that there is an aging effect. And then here's the Y-maze data. The one thing that I would like to point out is that we powered our groups by expecting that a lot of them would die due to old age. So we started out with very large groups. And also because we had to split them once they reached 22 months of age into a treatment and a control group, which means that error bars from 6 to 18 months are kind of like tame and okay. And after that, they tend to be bigger simply because the sample sizes have gone down due to mice dropping out of the study due to health reasons or death, and simply due to every group now having less mice in it because they're split across the treatment conditions. But what you can see here is a clear effect of age, genotype, and a sex and genotype interaction effect. And what I would like to point out is the 24-month data, the blue bar. These are the APOE4 carrier males who seem to be particularly bad at this task at that age. So the summary of the aging effects are that, yes, spontaneous alternations in the Y maze decrease with age. So we have an aging phenotype in cognition. And this seems to be more reduced in the risk model. Nest building as part of apathy is not so much affected by age or genotype, but the reactive grooming component was so. And that seemed to be a little bit more reduced in these humanized APOE4 mice, but that was so far only a tendency and not a confirmed statistical difference. Anxiety, we have a clear increase in anxiety behaviors across all tests with aging. However, no effect of genotype. So, well, what does this mean? Is this good or bad? I think that's in the eye of the beholder. These mice don't have aggressive Alzheimer's-like pathology similar to other mouse strains. They're mice, they don't have dementia. They're just aging mice who have some risk factors that are shared or have been introduced into them, which are borrowed from what we know in humans. So I think this model is holding up pretty nice so far. the question is, what happens with the drug intervention? Well, nest building didn't do anything for these mice before, and it doesn't do here either. The drug intervention doesn't really change anything. So we simply use this and say, okay, I mean, if a mouse doesn't make their nest anymore, I think something has to be seriously wrong. And this is kind of like a good thing that these mice are not really that severely affected. At 27 months of age, you can see that the quality of the nest has gone down a little bit, but not really that much, no precipitous drop. Well, the drug intervention decreased anxiety, but only in the open field test and only at 24 months of age and not at 27 months of age. So you can see here, we have significant sex effects, which is why we have all these colored bars here, which are a little bit difficult to navigate. But that's basically the best way to really show you these data. And you can see that, oh, the titles. Oh, yeah, only at 27 months of age, sorry, for the open field, was there a significant effect of the treatment. Other than that, the treatment didn't really change that much. How about novelty suppressed feeding? We saw a clear treatment effect at 24 months of age, which went away at 27 months of age. So at 24 months of age, the animals, all of them decreased the amount of time that it took them to approach the food. There is one little snack that I will mention later, but keep in mind that there may be an alternative explanation for what I'm showing you here. Cognitive testing, well, this is the most intriguing finding. We didn't see any effects at the 27 months of age cohort, but at 24 months of age, we saw a significant genotype and treatment interaction effect. And again, this seems to be carried by these APOE4 males who perform really poorly on the control chow, but when given the intervention, have a much improved behavior outcome. At 27 months of age, right before these mice are sacrificed for tissue collection, we do a last test. We do a memory test using fear conditioning. And the reason why we're doing this at the end, of course, is because it's a stressful test. And we don't want to introduce chronic stress. But if this is the last thing that is happening to these mice before we collect the tissue, we might as well. However, all we got was a significant effect of sex, suggesting that the males froze a little bit more to the fearful environment than the females. And that's that. Everybody basically showed the same level of freezing, the males being a little bit more fearful or having a better memory. I mean, that's the question here. But the drug really, unfortunately, didn't have an effect on this. So we see moderate improvement for cognitive behaviors at 24 months of age in the Y maze. Apathy, no effect on nest building. These splash test data are still under analysis. In anxiety, we see a mixed effect. It seems to be that we have decreased anxiety in open field at 27 months of age. It shouldn't be 24, it should be 27 here. And decreased anxiety in the other test, the novelty suppressed feeding test at 24 months of age. So does this intervention really help these mice to become healthier during aging? And does this result in less cognitive outcomes and mouse behaviors that correspond to some of the psychiatric symptoms that we see in human Alzheimer's disease? Only to a certain degree. Other observations that we made are related to survival. The interesting thing is that more females than males were removed from the study. They had to be euthanized due to health issues. A lot of times this was related to cancer, tumorous growth. And only in female APOE3 carriers did the drug cocktail actually reduce or increase the survival rate. As you can see on the right-hand side, the males fared much, much, much better than the females. I really don't know what to make of that. That's an unexpected finding. But that is what it is. But here comes an interesting thing. The drug cocktail results invariably in around 10% weight loss for males and females. So you can see here, on the left are the females, the ApoE4 carriers at 28 weeks after beginning the treatment, they're at 90% of the body weight that they had before the treatment started. The ApoE3 carriers don't have that much of a drop, but keep in mind that more of the ApoE3 carriers died, which suggests that they died before they could have a precipitous drop in weight. And these graphs are computed in a way that a dead mouse's weight is carried forward. With the males, however, you can clearly see no genotype effect, but all of them are leaner, presumably because of the acarbose, perhaps, resulting in less efficient glucose absorption in the gut. So they don't get as many calories out of their chow. But this could be a problem for a behavior test that uses food as a motivator, because if these mice weigh less, maybe they are hungrier. And maybe that's why they approach the food, not because it reduced their anxiety, simply because it increased their food exploratory drive. So far, so good. Now what, right? Are there differences in age-associated processes between these two mouse strains and in response to the intervention? So we have collected tissue from most of the mice. Some of them are still alive, so the study is still going on. A handful of mice are still in the study. We have collected serum, frozen tissue from brain, liver, kidney, spleen, heart, and lung, formalin-fixed liver, kidney, spleen, heart, and lung tissue. And we are really willing to share some of this tissue if anybody has a great idea. Our plan is to look for geropathologic lesions to see how damaged these organs really are. But obviously, we are not going to use up all these organs for these purposes. And one intriguing question is, so these humanized APP mice don't have any plaques. Nobody has ever reported any plaques for these. But most of the studies that were done in these mice were done like this. At most, maybe 18 or 20 months. Here we have mice that are 27-month-old. You add all the behavior testing. At time of death, they're easily 28 months. So maybe here you would have a mouse that could have plaques without having all these terrible familial Alzheimer's mutations. Could there be a vascular pathology angle? We know from published work from colleagues that these ApoE4 mice have some blood-brain barrier dysfunction at 12 months of age. So the question is, does rapamycin, with its effect on the vasculature, perhaps decrease the leakiness of the blood-brain barrier in these mice? So that would be following the aging angle. This is something that we do a lot with our collaborators in the Ladiges lab. And on the right-hand side is something that's coming more out of my lab. And this is, okay, let's take a look at the behavior data and let's follow the behavior data. Are there any differences between the two mouse strains and in the response to the intervention in brain regions that are known to be important for anxiety behaviors and spontaneous alternations? You can see here, there are a lot of brain regions connected in networks, and it may be worthwhile to look at the frozen brain sections that we will be generating from these mice to see if there are any changes in these brain regions. We have started this by focusing on the prefrontal cortex, which is a brain region that is important for spontaneous alternations in the Y maze and has implications also for the psychiatric conditions. And we took some RNA samples, sent them away for sequencing, and well, we're analyzing the data right now. So, but these mice don't really have that much amyloid pathology. So let's talk about mouse models to assess late life Alzheimer's disease neuropathologic change. So let's bring amyloid and tau into old mice. Instead of using mice that generate these molecules spontaneously, well, the ApoE, APP mice don't really do that. If you want to avoid familial mutations, you will not really get this. So we took advantage of adeno-associated viral vectors, AAVs, And new serotypes that have been shown, if you inject them intravenously into the retroorbital sinus, that these cross the blood-brain barrier and result in beautiful expression of reporter fluorescent proteins all across the brain. So we thought, oh yeah, this is great. Let's use old mice and simply a little poke here into the eye, retroorbitally, not really directly, just damaging the eye while you do this. That should do the trick. The big disappointment is that there was only minimal induction of amyloid and tau production in the prefrontal cortex and hippocampus, and absolutely zero changes in behavior. And I would like to contrast this with two other cargos that we are using in my lab in collaboration with other people here at UW, Nikki Liachko, Caitlin Latimer, Monica Sanchez, and Scott Kennedy here. And that is, one is called a mitochondrially targeted ApoBac that results in mitochondrial deletions. If you inject that the same way into young mice, it takes a while, but these mice develop a pathology that is really not compatible with life. And the other thing is if you overexpress TDP-43, which is another protein that is involved in neurodegeneration, then you see a very, very clear early onset phenotype right around 7 to 12 days after you do the injection, where these mice develop severe ALS-like symptoms. So it's not like this virus isn't really working, but it really didn't do for us what we wanted it to do. So like always, there are no shortcuts and there's no easy way out of anything like this. So we went and did experiments the hard way, where you do stereotaxic surgeries and inject these viral vectors that express a beta-42 and a tau version that gets hyperphosphorylated directly in the hippocampus. We do this in mice that had been getting the intervention at 22 months of age. For 2 months, these mice have been on the intervention. At 24 months of age, we perform the surgery where we do a cranial injection, and then we do behavior testing at 27 months of age. And what you can see is if you only focus for the first at the control chow part of these panels, you can see that if you express a beta-42 and this tau version, this p-tau version that we're using in the hippocampus, it clearly results in an obliteration of the spontaneous alternations in the Y maze, and it results in a significant reduction of freezing in the contextual fear conditioning experiment. So yes, when you inject these viruses directly into the brain, you see a clear effect. And the question is, would the chow help? And it does, indeed. That was the biggest surprise of them all. It seemed to completely reverse these phenotypes. That was really, really unexpected. And at the time being, we're waiting for more data to come in. We have collected brains. We have sent brain tissue to collaborators to look at proteomics changes. We're looking histologically at these to see if there's any clue as to why these animals really perform as well as they're doing here. So to sum it all up, yes, we have mouse behaviors corresponding to cognitive anxiety, behavioral symptoms of dementia that show age-dependent impairments. Some of them are dependent on ApoE genotype. And targeting of aging processes with our drug cocktail improves cognitive and anxiety-related deficits at 24 months of age, more so at 24 months of age, but not really that much at 27 months of age. Not all of these PHPEB AAVs are created equal, and these are these AAVs that I talked to you about when you inject them retroorbitally, should all cross the blood-brain barrier, but clearly that's not guaranteeing that your experiment will work. Induction of hippocampal amyloid and tau expression results in cognitive deficits. They can be ameliorated by the drug cocktail. And as I said, the molecular and histologic analysis are currently in progress, and we hope that they will yield mechanistic insight into this intervention, because if you go back to the beginning, it's basically a cocktail of 3 drugs that are very, very dirty in the sense that they target multiple processes. So who knows exactly what they're doing, and I doubt that it's going to be this or that molecule, but rather network effects. Well, not most of this, but some of this work is still work in progress, and most of this, if you talk about the molecular part. And my thanks goes to people in my lab, Daniel Fisher, Caitlin Schaefer, CJ Battaglia, Nivan Tran, and Emily Friedman. The last two are actually UW undergraduates, and both of them had posters at the recent UW Undergraduate Research Symposium for different projects. We have collaborators in the Keene lab. This has received a lot of support from Drew Keene. Amanda Kirkland was of tremendous help with some of the histology work that we are undertaking now. And of course, our main collaborator, Dr. Warren Ladegis, and our hippocampal brain samples are now in Devin Schwepp, he in Genome Sciences, in his lab to do the proteomics analyses. This is the last slide of the presentation, and this is where we take questions, and I hope have some kind of discussion. Thanks.
Yeah, I think you might have answered my question, but I just want to make sure. So the do you know the serum level of the interventions? Like my concern is like the acarbose, it prevents gut uptake.
Would it prevent also
Yeah,
uptake
does
of the other
does it
drugs?
mess with things?
Excellent question. We would have to take a look at that.
I mean, part of me goes, oh, maybe that's not an issue because you did if I understand the last experiments that you were showing,
Yeah,
that's the same intervention.
it's the same intervention, but you're right. I mean, the whole intervention, and this is the problem, could be carried by just one of these drugs. We don't know. Do you really have to take all three of them? Could acarbose be the one to take? And all the others are irrelevant because they don't really get. But acarbose is specific for the glucose absorption, so I don't think that the other drugs are.
I mean, it messes with the microbiome, which that can cause all kinds.
That's an excellent.
I mean, there was Alessandro shown that you just was it the butyric acid, I think, or whatever the
just a short chain fatty acid
yeah.
Could do the trick.
Yeah.
So the question is, and this is an intriguing question. This is why I think this is the time to really get the aging community here involved and say, okay, maybe what we need to do is the next set of experiments just has to take one of these drugs instead of taking the cocktail. But mind you, we took the cocktail because we just wanted to go for the biggest effect to see, is there anything to be seen at all? And we really didn't expect things to happen the way they happened, that actually the drug cocktail had that effect, which is still something that I'm scratching my head. And the next question really is, what exactly is it that's happening? And I hope that the proteomics analysis may shed some light on that. I think there's a thing in the chat. Oh, no.
So I wonder, so to give those mice medicine, you put those medicines in a chow, right? Yeah. So is this the best way to like deliver those medicines? Like how do you determine like the concentration of the medicine, whether like they actually?
Well, yeah, that's a great question, right? Because I mean, they're not humans, they're not taking pills. You cannot do injections into mice over a period like that. You will get inflammation. So putting it into the chow and making it orally available is really the best way to go. And these drugs have been chosen because they have already been compounded by the aging intervention program in mice. So it's based on that.
So did you check the, so on the last part of the study, so you inject the injection in the mouse hippocampus, did you check the pathology, do like staining?
We are doing that right now.
Okay.
So mouse brains have been sectioned, they're being stained, they're being scanned. This study, as you can tell, I mean, we're really following mice. We're breeding, we have bred mice, and then we have been following these mice for almost yeah, for 2.5 years, basically. So this took a very, very long time in preparation, a lot of logistics. So the exciting story, and I've done a lot of mouse behavior, so that's why I present you these data. The second large part is still coming, and that's the histologic analysis and the transcriptomic and proteomic analysis that may give us a clue what's going on, because I guess what you're hinting at is the question, is the pathology different in these mice, right? The pathology is driven by viral vectors. Would these mice be better at clearing up the pathology, or do you have less reactive innate immune cells to the pathology? Do you have a change in microglia, for example, or in reactive astrocytes to the pathology that is induced by the drugs? I mean, that's one of the things how, for example, a very recent intervention in Parkinson's disease is supposed to work by targeting astrocytes. GLP-1, I don't know how many of you have heard about that, but targeting reactive astrocytes has been shown to be beneficial in animal models of Parkinson's disease, and it made it actually into a phase 2 clinical trial with GLP-1 agonists. So that could be one way that this is going.
So my previous lab study Parkinson's disease, and I do some of those in-brain injections too. And normally we will have those mice after post injection, we will wait for like 3 months, at least 6 months. Then we started to see those pathology build up around 6 months. You will see plenty, and you only take a look at 3 months. I don't know about like Alzheimer's disease.
Yeah.
But I don't know, do you like try to probably inject the mouse at an earlier time point and so that you can incubate for longer?
That's a great point. To see really, I mean, one thing would be to say how much does it come into play, how long these animals are incubating the pathology. I think that's what you're asking. And the other question that you are having is the amount of time that we allow for this pathology to develop. I mean, you can see from the behavior data that yes, it's not going back. Okay, that the behavior deficit is there. And mind you, we didn't invent this AAV-based induction of Alzheimer's pathology. This is other people have used these viral vectors and have developed them. And their data suggested to us the time point that yes, we inject them at 24 months of age, we test them at 27 months of age. 3 months is usually enough to have some effects on behavior. And we saw very clear effects on behavior. Could they be worse? Of course. Could it spread beyond the hippocampus? That's an alternative theory. It has been shown that if you inject these viral vectors into a part of the mouse brain, they can actually induce seeding of pathology in non-injected areas. So I think you're making a really great point. But for our initial approach, we had to keep it simple. And these are all excellent questions for follow-up. Does this drug cocktail, for example, prevent the seeding of Alzheimer's like neuropathologic change? We know that amyloid and tau pathology in humans has a very specific pattern. And there are staging systems based on the distribution of these. And what if you had something that could put a stopper on pathology, tau pathology, progressing beyond the entorhinal cortex and hippocampus and sparing the overlying cortical areas? That would be great, right? But I'm getting ahead of myself.
This is sort of just a random question, but you mentioned the phenylbutyrate is an HDAC inhibitor. Do you just see increased histone acetylation across the entirety of the genome? Like, or are there specific areas?
Yeah, I mean, that's one of the effects. But as I try to kind of like make clear for all 3 of these drugs that they have multiple effects, that there is not just one effect that works, but the one that is the best characterized for phenylbutyrate is, yes, indeed, it's an HDAC inhibitor.
Thanks.
So you said most of the mice didn't make it to 27. Most of them die.
No, not most of them. I
No,
just said
some
with
of
the
them,
females,
yes.
50% of them died actually along the way.
They die of cancer.
Well, usually how this goes is that we get a notice from veterinary services. Your mice are very sick. It's the humane thing that you have to euthanize these. What are the endpoints that you have to find for the study? Usually it's weight loss, it's that these mice are very lethargic, that they're very, very sick. And a lot of times we can see that they have really masses on their body that you can feel, and sometimes you can see them. Yeah.
Yeah, I just wonder whether them having cancer will like interact with the results. Like,
Excellent question. I mean, we know that mice, one of the complicating factors in mouse aging seems to be that cancer seems to be more prominent in aging mice than it is in humans. So this may be simply an inherent limitation of the model system chosen. So we have to keep in mind all of these models and all of the data that we're showing, they're all limited. And I think you're pointing out a very nice limitation, or nice, a clear limitation of this.
thank you.
All right, thank you.